everyone, I'm Maddie, the research director here at the HNRNP Family Foundation, and I'm excited to welcome you to our very first Under the Umbrella series. We'll be trying to have educational videos to answer a lot of questions that many people have. And so today we're going to be talking about HNRNPs 101, which is just, what are the HNRNPs? And first, the HNRNPs stands for heterogeneous nuclear ribonuclear proteins, which of course is difficult to say. When we have the gene, we do it in italics, and when we have it as a protein, we have the lowercase hn and then the capital RNP. And what this means is it's a diverse group of genes or proteins that sometimes are in the nucleus, that bind to RNA and nucleic acids, and are proteins in the cell. And so these are what we look at all the time here. And each HNRNP is labeled from A to U, and the letters are just a designation. They don't mean anything in particular. So we have HNRNP C, D, G, H1, H2, K, R, Q, U, and UL1 and UL2 as some of the genes that we've been looking at the foundation. And the HNRNPs are an interesting group of genes because they do many things and also can do many things together. And so first, the HNRNPs are involved in in RNA processing. And so RNA processing is basically from the DNA all the way through to the protein. And the first step is spliceosome assembly. And so you may hear someone call an HNRNP-related neurodevelopmental disorder a spliceopathy, and that's because it's related to splicing, which is cutting up genes to make them into the version we're going to express. But the first step of this is getting the spliceosome together. And this just means recruiting a variety of proteins to the DNA, and they will then work together to begin transcription, which is DNA to RNA. And some of the HNRNPs that we know are important in this process are HNRNP-C and HNRNP-U. After we have the spliceosome all organized, we can start transcription. And so, like I said, transcription is from DNA to mRNA. And this all is occurring in the nucleus, which is what this circle represents. And so we start with our spliceosome at the top, ready to transcribe our gene. Once we have transcription involved, UL-type proteins 1 and 2 and HNRNP-K become involved. They then help the polymerase, which is that pol, make our mRNA. And once we have a mRNA, it starts as a pre-mRNA. And we know that multiple HNRNPs are involved in this step, including HNRNP-U, HNRNP-G, which is also called RBMX, HNRNP-H1 and H2, HNRNP-Q, which is also called SYNCRP, HNRNP-R, and HNRNP-K. And they can be involved in this in multiple ways. First is making sure that we splice the genes appropriately. And so splicing is the removal of introns, because DNA is made up of exons and introns within genes. And the introns are mostly regulatory. They're pieces that we don't need. And so the HNRNPs can help cut out those particular sequences and merge together the two exons, which is what we want for our gene to be expressed. And so once that occurs, and this can occur for a variety of genes, each HNRNP can target a subgroup of genes. Some of them can target the same genes or different genes, and they can also target themselves and each other. And so this regulation process is important both at just the HNRNP gene level, but also at a much higher level with all of the genes in the genome, essentially. So once we have our spliced mRNA, HNRNP-Q or SYNCRP, HNRNP-R, and HNRNP-C are known to be involved in confirming that that mRNA is correct. For example, HNRNP-C in particular is known to make sure the length is correct for each mRNA. And once we have our splicing complete, the mRNAs can exit the nucleus. And so when we exit the nucleus, We're now in the cytoplasm of the cell, and this is where we mostly can control gene expression, mRNA transport, and translation, which is from a mRNA into a protein. And with gene expression, one of the ways that we know HNRNPs are involved in this, other than controlling that splicing that we already talked about, is by degrading mRNAs that we don't need. So sometimes a cell might make a lot of something and we don't actually need that much. And so we know things like HNRNPD are involved in that kind of a signal. We know HNRNPQ or Hnrnp is involved in transporting mRNAs. And so sometimes mRNAs can just be made anywhere in the cell. Other times they need to be in a specific location. And this is really critical for neurons since these are long cells. And so HNRPQ and probably others can move our mRNA to the appropriate spot so the protein is made in a place that makes sense. And then finally, HNRNPs can be important in translational regulation. And so this includes HNRNPR, HNRNPC, and HNRNPQ, and HNRNPK. And each of these are really critical in making sure that the right mRNA is there and that all of the appropriate tools the cell needs to make these proteins are all in the right place, and will ensure that the protein is made appropriately. Transport is critical for neurons because those are long cells. And then I wanted to show you kind of what these HNRNPs look like. And so, like I said, there's a different letter associated to each HNRNP gene. And HNRNPC is thought to be the founder of the HNRNPs, in that it was found first in 1977. And then they decided that every next one they'd identify would be another letter of the alphabet. So that's why we get to be alphabet soup. We know that HNRNPC, HNRNPD, HNRNPG, HNRNPH, HNRNPK, HNRNPQ, R, and U are all involved in neurodevelopmental disorders. And we have several, such as HNRNPL, that are candidates for disorders. So we know some people who have variants in those genes, but we haven't confirmed that that is what's actually causing their issues. And you can see that some of these look more alike than other ones. And that is important in terms of both how these genes work and how they present as a neurodevelopmental disorder. And so, for example, HNRPH here is shown just as one protein. It's actually HNRPH1 and HNRPH2, which are encoded by different genes. And they look so alike that they just list them sometimes as just HNRPH. And that's proving to be really critical in the ASO trial that is going on with Yellow Brick Road Project in terms of regulation. We also know that the more these proteins look alike, the more the individuals who have variants in those genes look alike, which it can be somewhat literal in terms of their facial features, but also just in the neurodevelopmental features. And so some individuals with, let's see, which is a good example. Those with HNRNPQ and HNRNPR do have some shared features compared to some of the other HNRNPs. And I'm just going to go back to when we were talking about alternative splicing. And so this is one of the key things that HNRNPs are involved in. And this includes cutting out those excess introns, but it doesn't mean overlap of all these disorders. We know that most of them involve neurodevelopmental delay and intellectual disability, but there are certain smaller features that overlap with more related genes. And so these are things like the level of severity of intellectual disability, or hand and feet features, which usually are very mild, but they can still be genetically relevant. And it's just these small details that overlap that we see with these genes. They also tend to, if they look more alike, have a more similar behavioral profile. And kind of an example of this is that HNRNPC looks a little bit less like some of the other ones. And this actually is one of the HNRNPs that we have not identified very many people with autism in, whereas some of the other HNRNPs we have. And so that's involved, or that's related to how the proteins are actually structured, as you can see here. but also related to what genes that they can regulate, since each one can regulate its own subset of genes. All right. And then I wanted to really highlight alternative splicing. And so splicing is taking that mRNA, pre-mRNA to our mRNA by cutting out those excess introns that are mostly regulatory. We don't really need them. But genes can have different versions as well. And so we can see a gene that sometimes has an intron included and sometimes doesn't. And we know that HNRNPs are involved in both the actual process of splicing, but also choosing who gets to be spliced in or out. And when we have dysregulation of HNRNPs, we're still not sure yet how that's going to affect the whole splicing scene in the cell. But we're working on that. And we currently have RNA-seq studies ongoing, which is very exciting. And so soon we're hoping to have a better understanding of what are the gene targets of each of these HNRNPs and what happens when each HNRNP is altered. And this can include not having enough of an HNRNP or something like a missense variant, which we'll discuss in another video at another time. And so a target gene is where each HNRNP will go bind and control something. And so one example is the SMN1 and 2 genes. We know that multiple HNRNPs are involved in binding to that gene, and they can cause inclusion or exclusion of a particular exon, and that can be important in the development of spinal muscular atrophy. And so that's an example of where HNRNPs aren't causing anything negative, but they are using that target to then have a further impact. And each of the HNRNPs can choose which mRNAs or gene targets that it will bind to based on a particular sequence. So some of them will recognize poly-A's, which is just a bunch of AAA in the DNA. Some of them will recognize certain sequence like CGT. And it all just depends on how the DNA is made up. And they've evolved together so they can regulate similar genes if they need to. They can also compensate for each other in this. And so they can, if we lose one HNRNP, another one may come in and perform that same role. And so that is kind of the general gene target. And something important to note is that with the HNRNPs, we have variants in these particular HNRNP genes. But you can see that with their extensive involvement in many different processes in RNA processing, that a lot of different genes could be affected. And so we think that the different genes that each HNRNP targets will affect what the neurodevelopmental or the phenotype broadly is. And those that have similar or shared targets may have more overlap. And I know that was kind of a quick and dirty overview, but I think the main thing to take from this is that the HNRNPs, the heterogeneous nuclear ribonucleoproteins, are involved in gene expression. And this can be from regulating at the DNA level all the way to regulating at the protein level. And this is important because proteins are what make up everything in the cell. So without proteins, we don't exist. So we need regulation to make sure that we're getting the right protein. And the overall, I think, kind of goal here is to explain that gene expression is really important. And HNRNPs, and this is an example of how genes change over time. And so this is just looking at different levels of brain development, and it doesn't really matter what it looks like, but you can see there's different colors in different places. And that's just highlighting that there are different genes being expressed or not expressed. And depending on if there's an HNRNP available in that particular cell, that will change what genes are going to be expressed. All genes are regulated in some manner, otherwise it would just be kind of a free-for-all. And so the HNRNPs are really critical for not just the HNRNP family, but for a really large group of genes. And then, like I said, we think that changes in expression of those other genes is likely what contributes to some of these HNRNP-related neurodevelopmental disorders. I know that it can be a lot of information, And this is the simplified version, unfortunately. And so, definitely think on it. And any questions at all, feel free to ask. But thank you so much. Bye.